All right, welcome everyone. My name is Lynn Mullen. I'm a senior scientist with QIAGEN Bioinformatics. And today I'm going to show you how IPA's knowledge and discovery tools allow you to relate the most recent literature findings to your research, how you can create interactive and customized pathways, and how to leverage all of this information instantly without needing to upload any of your own data. This webinar is suitable for new users of IPA and also those of you who are intermediate, even more experienced users, and you want to learn how to use the different functions a bit more effectively. It's scheduled to last for 45 minutes, and I'm going to spend about 35 minutes or so presenting, and then I will open it up for questions at the very end of the presentation. You might have noticed that you are muted right now, but as we're going throughout the webinar, feel free to go ahead, type in your questions into that question box as we're going along, and that question box is located at the bottom of your GoToWebinar control panel. And we're also recording this webinar, and it will be made available to you later. And this is just a note that the software presented today is for research purposes only. This is the last webinar in this webinar series. So over the last 2 weeks, we held webinars on how you can format and upload your data into IPA and also how you can interpret a core analysis. And the recordings for these webinars will be emailed to you soon, if not already. But first, I'd like to provide you with a brief overview of the webinar. So I'm going to start with an introduction to the Ingenuity knowledge base, which contains findings that underlie every algorithm that the program performs. I'm going to show you different ways that you can start to use IPA both with or without data. And then I'll next tell you about some questions that we'll address today using an example. And we're going to do this with a live demonstration in IPA. And then finally, I'll answer your questions. Okay, so in the first webinar, I introduced you to the concept of the Ingenuity knowledge base. And now I'd like to go into a bit more detail about it. So to help you to easily and quickly test hypotheses, we've developed the best-in-class knowledge base. So this is essentially a database of information that includes a massive amount of millions, really, of manually reviewed findings from the peer-reviewed literature. What we do in these cases is we have PhD scientists and we have MDs that carefully read the literature, and they upload those findings into our knowledge base. We also have a staff of scientists who create models to better understand metabolic and signaling pathways. We directly link to public databases like miRNA, clinicaltrials.gov, among many others. And we also have experimental data sets that further allows us to gain more information. So for example, for the analysis match feature, we have about over 57,000 different experimental data sets that you can match with your analyses. And you can have your own version of the knowledge base where you can securely add data to this, and that can help you make some important connections with your research. Now, with all of this content acquisition, we also have a quality control procedure to make sure that the findings that underlie the knowledge base are accurate and timely. And in addition to simply giving you access to the content, we've structured this into something that we call the Ingenuity ontology. And this structure is how the analytical tools will use this information. So this combination of the data, the information in our knowledge base, and the software allows you to quickly and accurately interpret the biological meaning of your omics data. And the knowledge base is not static. So as science is constantly evolving, so too does our knowledge base. So it's updated every week. And currently it's as of the end of September, we have about 6.9 million findings. So what do I mean by a finding? we've structured the information that I just described in the earlier slide into what we call a finding. So they're pieces of evidence that support a relationship and a prediction in IPA, and we've structured it so it can be used computationally. So it consists of details of an experiment that was published in the literature or database sources. So here's an example of a finding that you can see when you're in IPA. So here it's been observed that in a specific mouse strain, a homozygous mutation in the ABCB4 gene increases cholestasis. And as you might have noticed in the first webinar, we've actually taken this and we don't just show you this information, and we actually create a visual diagram to make that information much more palatable, easier to view. And as you might have remembered from that first webinar, we also color code it, which makes it a little bit easier to immediately see these patterns. So this structure is what powers the algorithm for the analyses and how they can be interpreted in IPA. Okay, so now that I've told you about the structure and the findings and the knowledge base, I now want to tell you about the different ways that you can access this information in IPA. So there's several different ways that you can analyze the data and search and explore the Ingenuity knowledge base. So one way, of course, and we went over this in the first two webinars, is uploading your own data set and then going ahead and just running the different analyses that we have to offer. But we also have further information that you can use both with and without data. So I'm going to go over these different options today. So bioprofiler, we have a search engine, and you can also just start off by creating a custom pathway, and you can integrate all of this information. Today, we're going to take the approach of going in and creating a customized network, a customized pathway without any data whatsoever, just starting off with a question. Okay, so today, now what I want to do is provide you more specifics about the tools that we're going to use today to address certain questions. So here they are. So we're going to use an example of diet-induced obesity, and we're going to ask what's known about the molecular basis of this disorder. So we're going to use IPA to find different molecules and pathways that are associated with diet-induced obesity. And there's many questions that we could address today, so I'm only going to focus on a few because we don't have that much time today. First of all, we want to ask which molecules are associated with obesity, and specifically, we'll ask which of these molecules, when showing an increase in expression, cause a decrease in obesity. So we're going to use the disease search and the bioprofiler tools to answer these questions. And then we're going to ask what's known about these genes and how do they interact? And then how are these genes connected? What are their relationships? And then which pathways do they belong to? And so for this, we're going to use a bunch of tools called the build tools. There's a gene view page and a molecule and relationship summary. And then we're going to ask what drugs are known to target the genes of the pathway, and how would the addition of this drug affect other molecules and functions that are in this IPA canonical pathway. So for this, we're going to use some overlay tools to address these questions. All right, and with that, let's go ahead now and search and explore an IPA. So let me go to IPA. And over the course of the past two webinars, I've shown you specific aspects of this welcome screen. And today I'm just going to focus on those that I haven't mentioned so far. So I didn't get a chance to show you this Learning IPA tab that's here in the center of the screen in this Quick Start menu. So you could go ahead and look at different workflows and different tutorials. And of course, I showed you already the shortcuts where you can start your analyses and upload your data. Over here, over in File, you could go to New, and you can just go ahead and click on My Pathway to just start creating a custom pathway. And you can do the same option when you select the New button as well. And then over here on the left, we have our project manager, and I want to highlight right now the library section. So this contains if you have the analysis match license, you'll see all the omics off different data sets, but we also have all the canonical pathways that are listed here, and you can go into details and click on those. We also have some talks lists that are present in our knowledge base. Okay. And then finally here, we have our search engine. So you can search for different genes, chemicals, diseases, pathways, etc. So this is where we're going to start today. So I'm going to choose this diseases and functions tab, and I'm going to enter obesity. And you can see as I do this, there's an autocomplete menu that pops up. So I'm just going to go ahead and select that one. You don't need to choose the auto select. You can just go ahead and hit search, and it'll give you a lot of different options. And you can also, if you want to search for multiple terms, you can enter them, just separate them with the comma. So here you can see the matching diseases and functions, and this is assorted according to the Ingenuity ontology. So we've classified obesity as a nutritional disease. And you can see here that we have over 1,000 associated molecules with obesity. And when I click on this plus sign, you can see the list of all the different molecules right there. So today what I want to do is I want to select this, highlight this, and then I'm going to go up here into the toolbar. I'm going to click on Bioprofiler. So I'm going to go back to my slides while this is loading to tell you a little bit about Bioprofiler and why we're choosing this for this step of the webinar. So Bioprofiler allows you to quickly profile a disease, a phenotype, or a function. So you can use this tool to find molecules that are known to be causally related to a particular disease or phenotype. So for example, you can find molecules that could be potential drug targets, or you could identify targets of toxicity. So the tool gives you different filter options, and they allow you to explore and discover the fine-grained relationships that exist between molecules, diseases, and functions. All right, so let's go ahead and take a look at it in IPA. So this table that is generated contains a lot of information, and it's limited in this column, disease and function, to obesity. And you can see each of the molecules has its own row, and I have a mixture of different molecules, including chemicals. And what I'm going to do today, because one of my questions was I'm interested in which drugs could be used as targets for obesity, I'm going to go ahead and instead of just picking any gene, I'm just going to select particular genes that people tend to use when they're trying to look for drug targets like enzymes, different receptors, ion channels. I mean, I could pick a bunch of them, but I'm just going to stick with these transmembrane receptors, transporters. I think that's good. And then hit apply. So now it's processing, and now we're down to 413, as you can see in the lower left corner. And if you recall, I was wondering which molecules would show an increase in activity. So I can just click on that. You can see a lot of these little filter buttons as you're going through IPA. And then I want to know which molecules which showed an increase in activity will actually decrease obesity. So I'm going to select decrease here and click on apply. And now we're down to 11 causally relevant molecules right here. And you can see there's a lot of other things that you could filter for, whether there's known biomarkers, for example. Did the evidence for the relationship between the disease function and the molecule, which species did it come from? Are there known drugs that target it, for example, and more? Now I'm going to go ahead and highlight them, and once I've selected at least one, you can see that these become active. So the cool thing about Bioprofiler is you could also limit it just to a data set if you have a data set. I don't have a data set to work with today, so I'm going to skip that option. You can export it, create a new data set. You can add it to a new pathway. I'm not going to select that option. We are adding it to a new My Pathway, but instead I'm going to choose the display as network. And the reason for that is it will automatically make the connections from the molecules to obesity. And if I had just selected the add to my pathway, each one would only be a separate node and they would have no connections. So this is the My Pathway feature, and this is what allows you to, we have tools here to create customized pathways and networks for your molecules, your different biological processes and diseases, for example, that are of interest. And I'm going to go ahead and maximize the screen, and then I'm going to zoom it in a bit. Okay. So this is a fully dynamic network. We can move things around. And here I'm going to walk you through these three main buttons. We have the building our network. We're going to interpret some of the information from overlaying it. And then finally, what we'll do today is we're going to make a nice, take the pathway of interest and make it just look prettier for a publication. So one thing that I want to do is point out this change layout button. So there's a bunch of different options for how you visualize the molecules and diseases and functions here. So you could choose subcellular, for example, if you want to see how these molecules are, where they're positioned within the cell. I'm going to choose organic today and go with that. Maybe move things around a little bit. All right, so what I want to do now is address our question about whether and how these molecules interact with each other. So what we're going to do is we're going to select all of them. You can do this in a number of ways. You can drag your mouse over it. You can select Control A on your keyboard. And by the way, Mac users, you still select Control A, not Command A for IPA. And now we're going to go to Build, and we're going to connect. So select connect. We have a whole bunch of different filter options. So I could say apply, and I'll do that right now. So this little button down here, but I could also change that and say, you know, only show me connections that are from human information, or only show me connections that are between particular cell lines, like adipocytes or something like that. So I'm going to close like that. And so now you can see all the new connections that have been formed. When you click the background, it goes from blue to this fuchsia color, purpley color. This shows you all the new connections that have been made, and some people don't really like that color. So what we can do again is highlight everything. And there's this button up in the upper left corner called Graph Option. And so you can click on that, or you can unhighlight the selected. And so now it's back to the gray. You can also just right click on the canvas here and you get the same options that appear. Now, if you recall, I started this webinar series discussing a case study of gemfibrozil, and that's a PPAR alpha agonist. So I'd like to continue along those lines and focus on PPARA. So if I wanted to see what only interacted with PPARA, you can see as I click on it, it turns blue, and we can see these aqua lines. And so this is telling us these are the only connections that are being made between PPARA and everything else in this network. But one thing that I really like to use is this path tracer tool. It's up here in the toolbar right here. You can fade the distant nodes, and so everything that is not connected to whatever you've selected on the screen is going to fade out. And you can change the focus so it's 1 or 2 or 3 or more steps from or hops away from the node that you've selected. You could keep it upstream or downstream. You can make that fade more complete or less complete. Okay. All right, so I'm going to go ahead now and deselect that. And let's say that you don't know much about a particular molecule or disease or function in the networks, and you want to access the knowledge base and learn everything that you could possibly learn about that. What you can do is just double click on that molecule, and down here on the lower right, you can see I get this molecule summary. So this gives you information from EntreGene about its name. You can find information like, well, what are the microRNAs that target it? What is the Go annotation associated with this gene? And if you want to learn even more about that, all you need to do is click on this hyperlink for the gene name, it'll open a new page up in your browser, which I'll show you right now. And this is called the Gene View page. So this gives you information, everything that we have in the knowledge base about that particular gene. So a comprehensive overview of all the different findings. And you can also do this if you're looking for a chemical or a disease. So we have this overview that's kind of basically what you're seeing over here. And you can also see information like the isoform view. So if there's any known isoforms, those will be located here. The descriptions from the different databases, different drugs that are known to target it. And then we have this categorized literature findings. So if you're following along, from home or office, I actually already clicked on the show details because it takes a few seconds, and I want to spend as much time as possible showing you IPA rather than waiting for a browser. But you can see these headers up here which tells you information like the biomarkers, if there's any known biomarkers, the functional roles, mutant information. I'm just going to quickly scroll through it because I don't want to read every single line to you. I think you would be bored to tears. But I just want to point out a couple things. You'll see these numbers in parentheses, and this tells you the number of findings that are associated with that particular information. Like there's 881 findings indicating that a homozygous mutation is associated with these particular diseases and functions. And then we can continue scrolling to find the diseases, and we see obesity right here. And you can see all of these are blue. So yes, they are hyperlinks. And when you click on the particular hyperlink, then what you'll get is this finding. And you might recognize the structure of this finding from my earlier slide. So here's the information that we can find, and then you can access different databases, like MGI, for example. You can click on that here, or click on this PubMed ID. Or for this particular paper, it'll take you straight to PubMed, and then if you can go to the different papers if you want to read the full text of the paper. So here you can go ahead and you can read the evidence for why we're making a particular conclusion and convince yourself of that. Okay, so let's go back now into IPA. I'm going to go ahead and close this molecule summary. And now what I'd like to do is, I've shown you how to find the information about a gene using the gene view page. And now I want to show you how you can identify the types of relationships there are between the molecules in this particular network. So I'll just use the example of looking at the relationship between PPAR-A and PPAR-gamma. So you can see as I hover my mouse over the line, we have these different letters. So A, E, P, P, and T. When I double click on that line, now I get something else in the bottom right corner, and this time it's called the relationship summary. So we can see what's exactly is going on. So A stands for activation, E is expression, protein, and transcription. And then if you want to go ahead and go straight to all of these findings, you can click on this view relationships, and it will take you to another page in your browser that will show you all of the findings for these relationships right here. Okay, so now I'm going to go ahead and click OK. All right, so now what I want to do is I want to further explore and customize this network. And so one tool I'd like to show you is called the Grow tool. So I'm going to go to Build, and then now we're going to go to Grow. That's the first option. So this tool allows you to find and add other molecules or diseases and functions. And you can also add one of the newest things we just added a few weeks ago with our fall release was the ability to also add canonical pathways as their own individual nodes to your networks. So I'm going to give you an example of this. Well, again, we'll pick PPAR alpha as our example. And I'd like to find all of the canonical pathways that are known to be associated with this particular molecule. So what I'll do is I'll select grow out. I'll pick it's all molecules or canonical pathways. So we'll keep that as a default. Doesn't matter if it's upstream or downstream. It doesn't really matter. You can limit it to what's in the knowledge base. And then what I'm going to do is go to node types. So that's this little plus sign down here. I'm just going to select canonical pathway, and then I'm going to hit apply. And now you can see all the different canonical pathways that are associated that contain PPAR alpha. I'm going to again go to organic view to structure that a little bit more easily defined for us. So you can see there's information associated with obesity, right? Shouldn't be too surprising to see things like the white adipose tissue browning pathway or PPR signaling pathways. Okay, so I've shown you how we've identified genes that cause a decrease in obesity. I've shown you how they interact and how you can find pathways to which these different molecules belong. Now what I want to do is I want to show you how you can find which drugs interact with the molecules in this network. So what we're going to do is we're going to find those drugs and we're going to add one of them to one of these pathways. And then I'm going to show you how we can predict what effect that drug would have upon the pathway. So we're going to go from building a network to interpreting the data. So to find all the drugs associated with the molecules here, we're going to move from build and go to overlay. So there's a lot of different options you can use here, and you could look for canonical pathways here as well. When I click on drug, and it works really fast, so I apologize for that. I clicked on the drug, and you can see all of the known targets of different drugs listed here. So this first one is called bezafibrate, and it's known to affect a bunch of the PPAR genes right here. And when you select a box, that we see this little node appear, it says RX bezafibrate, and it connects to these three different molecules. Okay, so what I'm going to do now is I'm going to say, all right, well, what happens if we added this drug to a system? What effect would this drug have upon a particular pathway? So I'm going to choose PPAR signaling. And when you double click that, you get a new window that appears, and this is our IPA canonical pathway view. So what I'm going to do now, basically the canonical pathway view that I mentioned in the first webinar is just a diagram of the molecular interactions of the molecules, and it shows you that at the cellular level. And sometimes we'll also have different diseases or functions within that drawing of the pathway. Okay, so what we're going to do is we're going to go back to build, and we're going to go to add molecule relationship. I could really choose any of these. I'm just going to pick biologic drug. It doesn't matter really. I'm going to click on the screen, and now I have that little shape here. I'm going to double click on it, and now I get something that looks just like what we see up here for genes and chemicals. And I'm going to type in bezafibrate and do a search. And now we have one. And so you can see right now here I have families biologic drug. Watch what happens when I select it. It changes it to chemical drug. So it takes the information from the knowledge base and it applies it here. And now we're going to click OK. All right, great. Now what I want to do is I want to find out, well, what molecules in this pathway are being targeted by this particular drug. And I'm going to go back and use that connect tool. Now you might recall that when we used the connect tool before, that I selected everything on the pathway and we had connections with all the different molecules. And we could do that here, but that's not really what we want to do. For example, if there is something between CRAF and insulin receptor, I don't want to see that, really. I just want to see what connects bezafibrate to the rest of the pathway. So all I need to do is select bezafibrate and click apply. And here it's going to just show me what bezafibrate is known to target. So you can see that it affects quite a lot of molecules within this pathway. Okay, so now I want to address our final question. What would be the effect of this drug on the different molecules and diseases and functions in this pathway? So what we're going to do is we're going to go back into overlay, and we're going to use what's called the map tool. And let me go back to my slides for just a second to describe this a little bit better before I launch it. So the molecule activity predictor, or MAP tool, enables you to predict the upstream or downstream effects of activation or inhibition of different molecules or diseases and functions within a network or a pathway in IPA. So you can examine these relationships by starting, you can select a particular node that you want to start with, and you can say, this is activated or this is inhibited, and then examine what effect it has on a set of neighboring molecules that have a so-called known activity or expression or phosphorylation. So here I'm showing you a cartoon of a network of different molecules. So when I make the prediction using this paint bucket that this center molecule here is upregulated or activated, as shown in red, IPA is going to use different algorithms that make use of the knowledge base to make predictions of their relationships. So if you recall, the relationships have these little letters, icons, and then that will tell us whether any related molecules or diseases or functions are activated or inhibited. So the map tool allows you to see the overall effect so you can generate an accurate hypothesis. So let's go back into IPA and see this in action. So again, going to overlay, and now we're going to go to map. So you recognize this from the last slide. I'm going to go ahead now and click on that red paint bucket. So we're not activating bezafibrate, like it's not being upregulated or anything, but we have to just here pretend while we are activating it by adding it to the system, whatever that system may be, tissue, cell line, individual. And now what we're going to do is I'm going to keep it at upstream and downstream, but you can always change it to upstream only or downstream only. And I'm going to click Start Prediction. And let me get rid of this so we can see this a bit better. So now you can see this prediction legend that appears on the right side of the screen. You might recall the coloration from the first webinar, where orange means we're making a prediction something's activated, and blue is a prediction of inhibition. So now we can see the effect of bezafibrate if we added it to the system, how it would decrease insulin and TNF alpha, and how it would cause an increase in the PPR alpha, gamma, and delta, and also the effects that it would have farther downstream. So we can see that we have fatty acid oxidation and degradation. We're seeing uptake of glucose that's increased, and uptake of an increase in lipid homeostasis, and much more. All right. Okay, so I've shown you how you can start with a question, you can build a network, you can interpret this information, look for a pathway, add a drug to it, add a molecule to it. And now what I want to do is end by showing you how you can create a high quality figure that's suitable for publication. So what I'm going to do is I'm going to now select the Path Designer tool. And when I do this, I get this warning message. So basically it says, oh, you have predictions of activation or inhibition turned on. And once you're in Path Designer, you won't be able to turn those off. I'm fine with that though, so I'm going to go ahead and click yes to continue. So now we get a new window that appears. I'm going to maximize that and center it a little bit. Okay, so it has this, yellow gradient background. You may not be a fan of that, so you can always change the background color to something else. You can change it to a different type of gradient or a different type of color if you wish. You can do a lot of things with this. This would take another whole webinar for me to describe everything. Some things that you might have noted is we've added in a little strand of a nucleic acid there. Some of the shapes have changed from the general shapes that IPA uses to ones that are more biologically meaningful. And so you can do a lot of things. You can add a legend here. I can go ahead and I just want to show you really briefly how you can add some cell art. So I have this line to indicate the plasma membrane, but I'm going to delete it and instead go to cell art. And here's the cell membrane. And you just have to click on the background and it has these little things that you can get on there and stick it so it's somewhere with the receptors within the plasma membrane. So you can play around with that information there. And then once you've finished manipulating this, you can share it with a colleague who also has an IPA account. You can export whatever data's here, and you can also export this in a variety of different file format options and a whole bunch of different types of resolution. So for example, 300 dpi tends to be what a lot of different journals prefer. So we give you that option right there. Okay. All right. So again, I've given you just a taste of what the software program is capable of without data. And again, you can use the same tools with any results that you've generated from your different analyses. And I'm going to go back to my summary slides today to talk about what we have discussed in this webinar. Okay, so I'd like to end the presentation by just giving you a summary. I showed you several tools that we can use in IPA without data to get information that could potentially be pretty important for your research. So we searched and explored the knowledge base for information about the molecules that were known to be associated with obesity. So we used the bioprofiler tool to hone in on particular genes of interest. And then we looked for information about the particular disease and the genes that we saw in our networks using the disease search, the gene view pages, and the molecule and relationship summary tools. And in the My Pathway view of this network, we investigated how relevant genes connect with each other, and we found canonical pathways that are associated with these genes using the build tools. And we identified which drugs target these genes using the overlay tools, and then went back to the build tool to add this molecule to a particular pathway, and then use the map tool to make a prediction as to how the molecules and functions would be affected by this particular drug. And then finally, I very briefly just showed you how you can take all of this information and use Path Designer to create a figure that you can use for your publications. All right, so this completes the webinar, and I hope you have a greater understanding of how you can use some of the tools in IPA to search and explore the knowledge base. So I mentioned earlier that I'm recording this webinar. It will be uploaded to what I call our TV channel, and this is located at tv.qiagenbioinformatics.com. We have a whole bunch of different video tutorials that are available for you, as well as previously recorded webinars. And again, if you'd ever like to contact us, here are our phone numbers. We promise to give you a response within 1 business day, and often we do it before then. Here is the email where you can send different questions, concerns to ts-bioinformatics@qiagen.com. And if you're interested in a license, you want a free trial, you can go ahead and contact bioinformatics sales@qiagen.com. And then down here is our websites, including our TV channel. Thank you once again. I hope that this webinar series has been helpful to you. Good luck with all of your research, and I hope you enjoy using IPA. Bye-bye, everybody.
QIAGEN. Sample to insight.